Pleasure to have Peter Nigrovic here. He's actually in Seattle. We've all been meeting him in person, but we couldn't work out the COVID restrictions to actually have us sitting in the same room. So he's a few doors down, speaking virtually to all of you. So I first met Peter when I was a resident in Boston in pediatrics, and he was sort of a junior faculty member who had done this combined training in adult and pediatric rheumatology. So he treated patients with autoimmune disease across the spectrum of their lives. And then I left, and he just steadily advanced up the ranks, such that he is now most recently the chief of immunology at Boston Children's Hospital. And so in that role, he supervises multiple different departments, including immunology, rheumatology, dermatology, and allergy. So a whole 60 faculty are supervised by him. His research, I think, spans across a wide range of different immune topics with relevance to autoimmunity. And the particular one he's going to speak about is a cell type that I think we don't think about that often, but he's going to try and convince us that this cell type does more than just make platelets. So off you go, Peter.
Right. So, Sean, thanks so much for the invitation. It's fantastic to come. Fantastic to have the opportunity to speak with you about a topic that I assume nobody's working on, but maybe this will stimulate some thoughts about ways that you might be able to work on it yourself, or ways that we can collaborate. It's been really super to have a chance to meet people, including face to face. David and others, a wonderful dinner last night to sort of brainstorm about a whole bunch of things that we do and ways we might work together. But this is a topic that's sort of out of left field. I think it's certainly true for me as a rheumatologist, out of left field, to think about megakaryocytes as immune cells. So no disclosures related to this talk. So, we think of megakaryocytes as, I think we know that they're hematopoietically derived cells. They start as hematopoietic stem cells, differentiate through endomitosis into a large cell with multiple N in terms of the nuclei. They then make these long extensions called proplatelets, which are released in the circulation as these bilobed structures that shatter then into platelets in the circulation. So this is the megakaryocytes that we know as platelet sources. So when they do this, it's actually really quite beautiful. So this is a video from Joe Italiano, who's also at Boston Children's now. And this is a megakaryocyte filigreeing itself into platelets. Hundreds of platelets can be made by the same megakaryocyte. And I want you to just pay attention to that video. And what you'll see on that video also is that you have a cell that is one big round cell turning into lots of little round cells, which is very membrane intensive. And that's actually going to be important later on. So megakaryocytes are mostly in the bone marrow. We know that they're not a common cell, but they're prevalent in every marrow section that you see. They can also be found in spleen as part of extramedullary hematopoiesis. They migrate via the blood to the lung, and although there are not many of them, there's a lot of blood and a lot of time. And so it ends up being around 250,000 cells or more per hour leave the bone marrow and migrate into the lung, where they get stuck in the small capillaries and then filigree into platelets there. What proportion of platelets are made in the lung remains controversial. In the mouse, it said it's about 50% or so, but that's, I think, really very, very, very approximate. But it's true that in humans, if you look in the pulmonary artery, so the blood going to the lung, and the pulmonary vein, the blood coming back from the lung, the platelet count increases along that barrier, which is at least partly probably megakaryocytes making platelets in the lung. So this is a video recently from John Looney's group from UCSF showing this process. Here, this is a video of the pulmonary circulation. Green is platelets and platelet-related products. And you can see here appearing in the circulation and filigreeing over the course of 10-15 minutes is a megakaryocyte coming from the bone marrow, depositing the lung, and then shattering into platelets. So that's the process, that one known process that happens in megakaryocytes. So as a rheumatologist, as someone interested in arthritis, we became interested through a set of observations that were very serendipitous. And I don't want to go into this in great detail, but essentially we were interested in the mouse. We were interested in mast cells, and the there's a mouse called the WWV mouse, which has a compound null and point mutation in CIIT, which is the stem cell factor receptor. These mice are mast cell deficient, and they're resistant to arthritis by a serum called KVN serum, which is an IgG that deposits in the joints as immune complexes. But it turns out that that KIT, the stem cell factor, is also important in megakaryocyte development. And although you can restore arthritis in these mice by adding mast cells back, we found that you can also restore arthritis by adding megakaryocytes back, cultured megakaryocytes. And that these megakaryocytes give you back arthritis unless those megakaryocytes are IL-1 deficient, in which case they don't anymore. So we were able to show that megakaryocytes can make IL-1 in a systemically relevant way in the context of inflammatory arthritis. And how do they do that? They don't appear in the joints, at least anything that we've seen histologically. And we think that it's probably through the production of microparticles. So as you probably know, platelets, when they activate, they can release many microparticles, but megakaryocytes do them also. And this is from Joe Italiano and Rob Flamenhaft, two PIs in the Harvard system. And this is an EM of the edge of a megakaryocyte showing the microparticle production. These microparticles are both IL-1 and IL-1 alpha and beta rich, as we found. And then if you take megakaryocytes, culture them, and add the supernatant, which is the cytokines, or the microparticles purified, to fibroblasts like synoviocytes in this case, that either do or don't have the IL-1 receptor, you can look for an index production, KC in this case, of microparticles. If you add the microparticles, you can get production that's IL-1 dependent, in this case, IL-1 receptor dependent. And this is a positive control saying TNF activates them generally. Okay, so microparticles from megakaryocytes can make IL-1 and contain IL-1, and they can deliver it at a distance.
Peter,
How are they activated?
what's KC? I didn't understand that.
KC is an IL-8 equivalent in the mouse. So it's a neutrophil chemoattractant. In this case, it's just a marker of the activation of the fibroblasts, because when fibroblasts are activated with IL-1, here you can see here with the IL-1 just plain cytokine, they release KC.
Thanks.
And it's just a bioassay for activation of fibroblasts in this context. So how are they activated? We were interested in IgG as a possible activator, and we looked for FC receptors in these cells and found that megakaryocytes can express FC gamma R1, but not 2, 3, or 4, which are the other FC receptors in the mouse. And you can see that histologically as well if you flush bone marrow and then stain. And here's a megakaryocyte that stains for CD41, which is GP2B. We know GP2B3A is one of the important targets in platelets, and we'll use this a lot in this talk as a marker for megakaryocytes. So it turns out FC gamma R1 binds not just IgG, but also contraxin category innate immune stimuli. And so, in fact, you can show that these cells are activated via CRP and SAP, which are two contraxins made by the mouse, that to increase microparticle production, which is one pathway. We don't know for sure if that's the pathway relevant here, but it's one path by which they can be activated and sense systemic inflammation. So this I think was a big surprise to us, and we were not thinking about megakaryocytes at all. We as immunologists know that platelets are involved in immune function, that they can release microparticles, cytokines, mitochondria, they attach to neutrophils and other cells, and they've been implicated in multiple diseases. So in this way, it's not surprising that megakaryocytes participate in immunity. But in other contexts, with this, we now see that microparticles are also important, and there's multiple ways that they can be relevant. Others, John Semple's group in Sweden has shown that microkaryocytes can present antigen, including cross-presentation, and cytokines are also production. So that these are all things that megakaryocytes do in terms of immune function. But we want to go explore further and think about what else megakaryocytes do in immunity. And that's really what I want to talk about for the rest of this hour. And so we encountered this observation. So this is the bone marrow of a child with systemic juvenile idiopathic arthritis. And we know sometimes these children get macrophage activation syndrome. So you look in bone marrow to look for hemophagocytes. And we saw this cell, or cells like this. And this is a known phenomenon reading about megakaryocytes. It's called imperapolesis. This is not a hemophagocyte. This is a megakaryocyte with multiple cells inside it. In this case, CD66B is a marker for neutrophils, so some of these cells at least are neutrophils that are within the megakaryocyte in this confocal section. And this is a term that was made up in the 50s, actually, coming from Greek, M inside, peri, around, and polemi, wander about. So it's inside, roundabout, wandering. That's what the term means. And it was described in megakaryocytes for the first time in this paper by a Canadian physician scientist who happened to be looking at a wet prep of bone marrow and was fascinated to see megakaryocytes with these cells that looked like they were sort of moving around, neutrophils moving around inside the cell. And this is a still photograph of this. But it really had otherwise not been much investigated as to why this should happen. But it turns out it happens in every species that has megakaryocytes. So cows, mice, rats, rabbits, monkeys, humans. So it's evolutionarily preserved across 50 to 100 million years of divergent evolution. And so it's sort of an interesting observation that probably needs an explanation. So we decided we were going to study this. And the work that I will be presenting is really the work of Pierre Kunin, who joined my lab a while ago and has been just a phenomenally talented experimentalist and thought partner in thinking about megakaryocytes. So we did a simple thing, which is look in the mouse and look in cross-section of bone marrow. So this is bone marrow here, and you can see megakaryocytes with cells inside them that have a neutrophil-like morphology. And we looked in the context of our favorite model, which is caveat arthritis, and saw that enteropolesis was observed in about 2 to 3% of megakaryocytes at baseline, and that about doubled with inflammation. With samples from some collaborators, we noted that in cecal ligation function went up even further. So it was a pretty impressive, but still relatively infrequent phenomenon. Although the phenomenon here with multiple cells in the same megakaryocyte was observed only under stress. So to understand this better at a three-dimensional level, because sections are only 5 microns and megakaryocytes can be 100 microns in diameter, Pierre developed a system in which you inject in the living mouse a marker epithelium, and then you shave down the bone edges after you sack the mouse, and then you can image in Z-stacks to get a three-dimensional image of the megakaryocyte in the bone, connected to the bone marrow. And so you see here a megakaryocyte pressed up against the sinusoid in the bone marrow. And here's the megakaryocyte nucleus, blue is DNA. And you can see here nestled within this megakaryocyte, nestled actually very close within the nucleus, which we've seen multiple times, but don't really fully understand, a neutrophil stuck inside the megakaryocyte. And this, we know it's a neutrophil because it stains for Ly6G. So using this, we were able to quantitate it better and measure in 3 dimensions how common this is. And it's more common, it's about 9% of megakaryocytes in a resting state and about 20% of megakaryocytes at any one point in time by 3-dimensional imaging. So we developed an in vitro model, which was fortunately very simple to do. We just culture bone marrow in thrombopoietin, which gives you megakaryocytes after density gradient purification. So you get a pretty nice purity of megakaryocytes, and then you just add bone marrow to it and let them sit overnight. And what you get then is this is a single megakaryocyte here bordered by CD41 as its membrane. And you see this megakaryocyte, not all of them do this, but about 30% of them do this, take up neutrophils, principally neutrophils, sometimes other cells, but almost always neutrophils, which is what you see in vivo. And I just call your attention to the fact that there are neutrophils that look like they're surrounded by CD41 membrane and neutrophils that do not have any obvious CD41 staining around them. So the question is, where are those? So we followed this up by electron microscopy, and we see what we think would be the standard expected version, which is here's a megakaryocyte, here's a neutrophil within it. It's in a vesicle that we call an imperisome. So the neutrophil's nicely nestled within it, like you might think a phagocytosis cell would be. But then sometimes you see them more like this, where the neutrophil's closely opposed. Here's the megakaryocyte, here's the neutrophil, here's the bilipid membrane neutrophil, and that is at the megakaryocyte. And the membrane seems to train track. It sort of closes to becomes very tightly joined with that of the megakaryocyte. And sometimes we see these structures, we still don't know what they are, where the membranes seem to get fuzzy. The border seems to get fuzzy between them. And then I think the really remarkable thing happens, which is all of a sudden the neutrophil appears in the cytoplasm. So this is the megakaryocyte cytoplasm with its organelles. This is a single membrane, which is the neutrophil membrane. So now we have a neutrophil in a place that when we first saw these images, we didn't think a mammalian cell was allowed to be. So we have an idea that pathogens penetrate into cytoplasm, but that other cells penetrate into cytoplasm was new for us. And we've since done lots of other EM images, and we see sometimes the neutrophil emerging from the perisome. The perisome border, it seems to penetrate through it, which is how we think this happens. So we think that's what this is. So here's neutrophils in the perisome, and here's neutrophils in the cell cytoplasm. So this is not a terminal event for either cell. This is an early video. So here's now the neutrophil, the megakaryocytes in red, the neutrophils in green. You see it sort of looks like it's pushing its way in because it's almost got podocytes sort of reaching forward, but then that cell leaves. So it's not a terminal event. The neutrophils go into megakaryocytes and then they come out. And so this process is mediated at least in part through integrins. So this is a confocal immunofluorescence microscopy experiment of a megakaryocyte and neutrophils. Neutrophils stain for CD18 or beta-2 integrin chain. And the megakaryocytes do not have that chain, but they do have a counter ligand called ICAM1, which you barely see at all unless a neutrophil touches the cell, in which case you see the ICAM cluster, and then a signal transduction molecule or a bridging molecule between the cytoskeleton and ICAM1, Ezrin clusters at the same space. And you can see here a neutrophil nestled up against ICAM and Ezrin in this context. And what's interesting is that this process is if you have neutrophils that lack CD18 or megakaryocytes that lack ICAM1, the process of imperapolesis is partially but not fully inhibited. That is, they are involved in the process, but they are not completely essential. And there are other molecules that we have not discovered yet that are involved in the process. So the actin is involved, and here's a filamentous actin stain where the megakaryocytes, where the neutrophils that are in imperisomes have an actin sort of shell around them, and that goes away when they actually enter into the cytoplasm here. And what's interesting here is that if you paralyze the actin cytoskeleton of the megakaryocyte, so it cannot actively phagocytose, you impair imperapolesis. But similarly, if you either using here leutrophilin or cytoclasm D, but if you do the same thing to the neutrophil cytoskeleton, it also happens. So this is an active process, both by the megakaryocytes and by the neutrophils. And that was the sense you got from that earlier video, that the neutrophil is not just a passive bystander, it is pushing its way into the megakaryocyte. So I think this raises the question, why? What would be the advantage of a neutrophil going into a megakaryocyte at all? And what would be the advantage in particular of going into the cytoplasm of a cell, where we don't normally think cells ought to be? So our clue here, I think, came from the live imaging. So again, this was this video that I showed you before. Here's the neutrophil with its podocyte sort of seemingly pushing its way in. And now here, 12 minutes, 16 minutes, 20 minutes, it's pulling its way out, and now it's almost entirely out, but it's left some of itself behind. And these are, in this image, the neutrophils are stained with a dye called PKH67, which is a membrane lipid stainer. So it's the membranes that are staying behind here in the megakaryocyte. So that explained another set of images we had that Pierre had generated, where there's a large megakaryocyte, here's some neutrophils within it with this PKH67, and it looked to us like the lipid dye was diffusing within the megakaryocyte. But lipids don't diffuse in aqueous media. So this is lipids going in different places. So something, again, lipids somehow are being left behind. And how that happens emerged from closer look at some of the EM images. So as I showed you in that first video from Joe Italian, when a megakaryocyte splits into multiple platelets, it needs a lot of membrane and it needs it fast because the surface area, of course, of the many small spheres is much, much larger than that of the big sphere. And the way it does that is to make a membrane reservoir called the demarcation membrane system. And so here's a megakaryocyte, here's the demarcation membrane system, this kind of filigree of lots of extra membrane. And it turns out that the neutrophil migrates over to the DMS and actually makes membrane fusion with the DMS and starts to feed its membrane into this system. So does it also transfer proteins, surface proteins? To address this question, we surfaced biatylated neutrophils, fed them to megakaryocytes, and then stained the resulting cells with streptavidin to show whether we can see neutrophil proteins emerge here on the cell surface. And in fact, you do see that. So here is a megakaryocyte. Megakaryocytes, this is the streptavidin staining. These are neutrophils in blue. And here blowing up this image, you can see a neutrophil. You can see it's got red around it because it's got it's streptavidin labeled biatylated proteins on its surface. But also those proteins seem to have transferred to the megakaryocyte because the megakaryocyte also stains red on its surface. But those, for example, that don't have neutrophils in them do not stain. So there's a transfer of proteins from the surface. We don't know what they are. They're the usual suspects like Y6G or beta-2 integrins don't transfer. So we're still trying to figure out what all goes from one to the other. So does this translate into transfer of neutrophil membrane to platelets? So megakaryocytes need flow conditions in order to make platelets. That image I showed you was not in flow, but most of the time, that was a really lucky image. Most of the time, if you just let megakaryocytes sit in culture, they don't make a lot of platelets because they need the flow shear stress to make it. So to make platelets, we use the mouse. And so here we made megakaryocytes and loaded the cytoplasm now, not the membrane, with a dye called CMFDA. And then fed these cells, bone marrow cells, where the membrane was labeled with cell view maroon. And peripolis is allowed to happen, and we hypothesized that now that there's a transfer of some of this membrane and therefore also the stain to the demarcation membrane system. And these megakaryocytes are injected then into mice. And so then we look by flow cytometry for platelets that are green, that is, they come from the donor megakaryocytes, and they're also red, which is to say that they have membrane from the donor neutrophils. And so we gate platelets in the usual way. So platelets are smaller. Here, this is a log scale scatter plot. And then CD41 is a marker of platelets. So you look for CD1 positive cells. If you feed the megakaryocytes in without feeding them neutrophils first, the platelets emerge like where you'd expect them to be. That is, there's no cell view signal. They're same as this is the endogenous platelets. These are the platelets produced by the injected megakaryocytes, which is to say they're green. But if you have fed the cells, these are fed unstained bone marrow cells. But if you feed them stained bone marrow cells, where the neutrophils have red on their membrane, you see that most of the platelets that come from the donor megakaryocytes have the transferred membrane. So it's pretty convincing that there is actual transfer of lipids from the marrow cells to the platelets. Or otherwise an in vitro system? I think that's a fair question. So to address that, we made a kind of a unique So we made a unique Chemeric mouse. And so here we lethally irradiated a mouse and put in two bone marrows. One is wild type bone marrow, which has no fluorescence intrinsically. And the second was bone marrow from a mouse that maybe some of you have used called an Mtmg mouse. This is a membrane tomato membrane GFP mouse, which when exposed to CRE turns green, but when not exposed to CRE, the the cells are labeled red. And what's unique about this is that the label is on the membrane. It's not inside the cell. It's not a cytoplasmic label. It's attached to the inside of the membrane. So here we can measure membrane transfer. So now we can graph these two and we sample the blood. And there are two possible outcomes here. One is if there's no transfer. So if all platelets are made either by these cells or by these cells alone, then we'll see platelets that are unstained or platelets that are stained, but nothing in the middle. By contrast, if there's membrane fusion in the process, then we'll see some population. We don't know how big here in the middle. Now, we won't see, of course, cells that transfer neutrophil and megakaryocyte from the same color. Right? We can't, those will be invisible. Similarly, red to red will be invisible, normal to normal will be invisible. Normal to red is we're not going to detect those either. It's going to be like on this end of the red curve. The only ones we can see or hope to see is neutrophils that are red transferring to megakaryocytes. So we expect to see maybe 1 out of 4 events. And in fact, we do see those events. So this is a staining of the resulting platelets. So this is a TD tomato and an anti-TD tomato to amplify the signal. This is permeabilized so you can get to the label. And here you have most of the platelets are made either by one marrow or by the other marrow, because either they're red or not red. But there is this hybrid population in the middle. And if you flush the bone marrow then and look at the megakaryocytes, you can see megakaryocytes that are either from one donor, from the TD tomato donor. This is CD41 in green, so red plus green makes yellow. So these are from the Mtmg donor. This is from the wild type donor. Those are green only. But here, we're lucky enough to capture a cell from the red donor in a green or non-normal neutrophil. And here, if you just look at the red signal, you can see that this cell is bleeding its membrane into the megakaryocyte, just like we saw in the in vitro setting. And so we call these here, so that tells us that in the normal setting, it's of course, it's an irradiated engrafted mouse, but in as close to the normal setting as we can come up with, there is a population that we call hybrid platelets that have megakaryocytes, that have neutrophil membrane on the platelets made in vivo. So we think this is at least 1 to 2%. So it's this number times 4, because we only see 1 out of 4 events. And then we think it's at least because actually the label staining is very thin. So we think that we will probably miss a lot of events. But so we think that there are, and we suspect that there are in humans as well, a population of platelets which was not suspected, which we call hybrid platelets, that have neutrophil and potentially other cells, membrane and proteins, likely proteins on them. So why would you want to do this? And I think we have only a partial answer to that. But one of the things that it seems to help is to make platelet production more efficient. So Joe Tagliano, who I showed you the image from earlier, has a nice system to study in vitro platelet production, or so-called proplatelet production, these long strands that come from megakaryocytes, that are put in culture and then the camera is visualizing them inside an incubator. And so over time, you can see the number of megakaryocytes that make proplatelets is increasing. And if you add the megakaryocytes alone, that's the black bar. If you add a bone marrow supernatant, you actually go down a little bit for reasons to understand. Fixed bone marrow cells makes no change. But if you feed them live bone marrow cells, then platelet production actually becomes more efficient. So this may be, although it's hard to prove, because you are able to get another source of making of membrane to assist with this membrane intensive process. So again, it's a reasonable question to say, is that true in vivo?
Peter, how do you control for like cytokines that the bone marrow is delivering?
So the idea with cytokines was the bone marrow supernatant here. So if you just take the supernatant from bone marrow, which would contain the cytokines, then you would expect to see, if this was all that, you expect to see the red bar up here somewhere. Now, you could say, what about the neutrophils interacting with the megakaryocytes and therefore making more cytokines? I can't address that problem. That could be the case.
Or other cells.
Or other cells, but something in the bone marrow supernatants. Something that's already living in the bone marrow supernatant is not the answer, because the supernatant doesn't do anything. Or actually it does things in the opposite direction. So I think we then asked, does it happen in vivo? And to address this question, we again use this cytoplasmic labeling system, where megakaryocytes cultured ex vivo are either labeled with green cytoplasm or with red cytoplasm. And then one of the populations, and we've swapped it in experiments, gets fed these bone marrow cells. So one has the opportunity to do enteropolesis, and the other does not have the opportunity to do enteropolesis. And then when we mix these cells together, we can then identify the cells from one and the cells from the other. And then we look in the mice to look for the platelets. So who are the platelets coming from? Are they coming preferentially from this or preferentially from this? And the answer, they're coming preferentially from the ones, this is over 2 days, platelet production, sampling the mice serially to see what proportion of platelets come from which donor. The enteropolesis co-culture system, it's not just enteropolesis, but that does seem to increase the efficiency of platelet production, and that effect is clear. So we published this as a sort of the first sort of functional characterization of enteropolesis in 2019, and building the following model. Here's a megakaryocyte, here's a neutrophil. The neutrophil interacts with ICAM1 and therefore is on the megakaryocyte. All their receptors involved as well. Using Ezrin, it binds to the actin cytoskeleton, and there's actin is involved and somehow in the process of the endocytosis of this neutrophil into an imperisome. The actin goes away, the vesicle seems to collapse around the neutrophil, and somehow the neutrophil then escapes into the cytoplasm, where it interacts with the demarcation membrane system and passes its membrane onto platelets, and then it leaves. Okay, so I think this raises just lots of questions, but including what is the fate of that neutrophil? And so from here, I'm going to talk to you about unpublished data, which I think is always more interesting. So in this video that I showed you,
Peter,
yeah, please.
Peter, I have a question. What stage of neutrophil is that? As you know, neutrophils can be staged in terms of their differentiation. Perhaps you'll address this. I'm just curious, is it a late stage neutrophil, an early stage neutrophil, because it happens in the bone marrow?
So we have not, so we're interested in neutrophil staging. We're interested in neutrophil differentiation, but we have not been able to figure that one out yet. It is the mature megakaryocytes that do it. That we've, by taking different days of megakaryocytes, you can see that the mature megakaryocytes are the ones that can do this. They're also bigger. The neutrophils all look like they have mature neutrophil nuclear morphology. So it's not the really early stage cells that do it.
So they're capable of all neutrophil functions. They have all the granules, everything's present.
Well, we can show that we can take neutrophils from bone marrow, which most of them are not really fully mature. So what we showed in a recent paper in Nature Communications was that there's a gap even between the most mature neutrophil in the bone marrow and the least mature neutrophil in the blood. There's a little jump, and we think that happens when the neutrophil transitions into the blood somehow. But we can show that neutrophils, if you harvest them, for example, from an inflamed peritoneum, they do this as well.
Okay,
so it doesn't require the cell to be immature.
Thank you.
You do it with the neutrophils from blood. Yeah. So it's not obvious that one is more efficient than the other, but we haven't rigorously examined that question. So here's again this wonderful video that I showed you earlier, where I highlighted here in the megakaryocyte that there was some neutrophil membrane left. But here, if you look at the neutrophil, it's got some megakaryocyte membrane because these are megakaryocytes are also stained with a lipid stainer. So what's going on here? Neutrophils are taking parts of megakaryocytes with them when they leave. So we went back to the EM images, and here's the neutrophil in an imperisome, and here's the megakaryocyte. And in the imperisome, sometimes it's pretty empty, but sometimes it's full of these exosomes, which seem to be coming from these multivesicular bodies generated by the megakaryocytes, because neutrophils don't have them. So here is, again, what looks to us, here, this is all still images, so you make of it what you will. But, you could sell me the story that this is a multivesicular body fusing with the imperisome to release its contents upon the neutrophil. And you can see the neutrophil then looking like, at least, it's entertaining the prospect of taking up these little particles because its membrane tends to sort of get all roughly in this area, as though it's doing micropenocytosis. So it looks like there's uptake of MK exosomes by neutrophils. And you can actually see this in culture systems. So these are stills from a video that you do in a simple culture. Here's a megakaryocyte, here's a neutrophil. We watch this one, and it migrates over to the neutrophil. And megakaryocyte, it enters, and very soon after entry, it starts to assume these little green dots, which are dots, CD41 positive dots. So that's a membrane-associated protein. And then as that cell starts to leave, it takes those dots with it. And in fact, you can see this happen. You can see this in vivo in circulating neutrophils. So again, this takes advantage of this Mtmg mouse, where again, everything's usually red unless it sees CRE, in which it turns green and stays green. So we took these Mtmg mouse, crossed them by PF4, which is a fairly specific platelet megakaryocyte CRE. We then we'll then have a mouse that has megakaryocytes and platelets with green membrane, and all other cells are red. Again, remember, this is a dye that's not in the cytoplasm, it's on the membrane, on the inner leaflet of the membrane. And then we did peritonitis. And if you use a PF4 CRE negative mouse, these are the controls, you see that the neutrophils in the space are red, they're TD tomato red, but they're not green. So this quadrant up here is empty. But if you harvest them from the mouse that has MF4 PF4 CRE, now you see that these neutrophils, which were red, also have some of them have something green in them. And you see this harvested preferentially, not so much in the blood, but in places, you do see a little bit in the blood, but you see more in places where they're from inflamed fluids. So these are the examples from sorting these cells, what they look like when they have the green dots in them. They look just like the ones we generated in culture compared to the ones that are GFP negative. So here's us. You can see this also in sections from these animals. So this is an inflamed omentum from a mouse where LPS was injected into the peritoneum. Here are lots of neutrophils, of course, in the peritoneum. Here's a blood vessel, and here's one that's actually in the omentum itself. And you see this neutrophil has all these green dots in it. And by contrast, the ones we happen to capture here in the blood vessel don't. So, or at least don't so obviously. So a hypothesis that I think is a reasonable hypothesis is that these uptake of this material, or at least something like it, is influencing megakaryocyte, neutrophil function. And so we purified exosomes from lysed megakaryocytes, did ultracentrifugation to purify them, showed by EM that they express CD63, which is an exosomal marker, and then did mass spec on the proteins. And we see lots of different proteins in this context, some cytoskeletal, some metabolism, some signaling. But we certainly see a number of interesting immunological markers, including things like PF4, TGF-beta, things that have neutrophil relevant functions. And if you take these megakaryocytes and put them with neutrophils, and then separate the neutrophils out and let the neutrophils migrate, you can see that compared to here along a transwell system, with LTB4 at the bottom, neutrophils that are incubated with live megakaryocytes do migrate better than those incubated with megakaryocyte supernatants or with fixed megakaryocytes or with no megakaryocytes. So it does seem like there's something going on in terms of migration. So can we visualize this? And so we took a system called a microslide, which gives the opportunity to put some kind of gel in the middle between two chambers. You can feed things to the chambers via these little holes. So we put marrow cells in one chamber, a chemoattractant in the other chamber, and here a collagen matrigel matrix with megakaryocytes embedded within it. And now you can watch them migrate from the side they come from to the side with the chemoattractant. Here's a chemoattractant gradient. And so I'll call attention to this megakaryocyte, which seems to get a lot of love from the neutrophils. This is a confocal image. So when you see them overlapping, they're actually going through. So let's start it up here. So here's the neutrophils. A lot of neutrophils just sort of passing through this megakaryocyte. You can watch those. And in watching them, then you can see that the ones that come out now have these dots in them. And the ones that are going in never have the dots. So you can watch them, the ones that pass through this, as opposed to the ones that pass around, develop all these little red, little green dots. And so those ones that have the dots actually migrate faster than those that don't have the dots. So here we used a TrackMate plugin on an image data to look at the speed of each individual neutrophil and characterize the ones that had dots versus those that didn't. Now, it doesn't say that we can't tell. It could be that those that were faster went into the megakaryocyte and therefore got dots. We can't say it's causal here. But in any case, we can say that there's an association between neutrophils taking a transmegakaryocyte route and faster mobility. So does this effect work in vivo? So again, this is one of these Pierre Cunin experiments that are just lovely. So here, what he did was to label bone marrow cells either red or green, culture them with megakaryocytes or without, and then mix the neutrophils together into 1-to-1 ratio, stick it into a mouse that has been given peritonitis, either LPS or IL-1 beta. And so then you can compare the neutrophils that are red versus green, that is, have had enteropoiesis opportunity, or at least megakaryocyte co-contact opportunity, versus those that have not. And you can see in this context that if you harvest from peritonitis, you can see that the ones that had contact with megakaryocytes do migrate with greater efficiency than the ones that did not. Sort of a subtle effect, but it's a clear and reproducible effect in 2 different models. So it does seem that there's a pretty clear effect from megakaryocyte enteropoiesis on neutrophil function. But is that the only kind of enteropoiesis that there is? So here we had a German medical student named Frank Huang in the lab, and he did a lot of serial video imaging of enteropoiesis events, as well as serial EM that I won't show you. And what was interesting was he observed that there was actually a pretty clear bimodal distribution of cells that went through really fast, that usually within 10 minutes, and cells that stayed. And so here, for example, is an image that in 10 minutes, it's going in, the neutrophil's going in, and then it's going out. And here's one that we call it fast enteropoiesis. And here's one that we call slow enteropoiesis, which just sits there. And many of those cells actually have sort of what looks like some kind of intimate contact with not actually the neutrophil membrane, we think, by EM, but with the perinuclear ER, which we think is really interesting, but we have no idea. So can neutrophils then use this way to exit the bone marrow? So in fact, that may be the case. So can they transit through this to get to the sinusoid? And we sometimes see events like this. So here's a megakaryocyte. Here in this 3-dimensional image is this neutrophil stuck against the sinusoid. So somehow it's going one way or the other. And again, you can measure that, look at that in 3 dimensions. So here's a sinusoid, here's a megakaryocyte, here's the interface, and there's a neutrophil stuck at the interface between the sinusoid and the megakaryocyte. So we imagine that that cell is going one way or the other here. So in fact, we're just building a 2-photon core at my division, but we're not there yet. So we work with several different groups that are better at this than we are, and we're able to visualize using 2-photon microscopy neutrophils in green in the marrow versus the blood, the sinusoid, and we're able to see that these events happen, and that about 15% of neutrophils seem to be 15% of megakaryocytes seem to be passing their neutrophils from the bone marrow to the blood on the way out. So we think that this is a separate function where the neutrophils are coming out different. We've only begun to scratch the surface of what those differences are. But I think this introduces a really interesting topological problem. So the neutrophils survive, the megakaryocytes survive, and yet somehow the neutrophils puncture its way out of the megakaryocyte. So how do you do that topologically? And so this is one of these images from the matrigel system. So neutrophils are coming from over here, passing through a megakaryocyte. This is more blown up. This is one of the earlier images where we had a lot of debris, so please forgive. But I think that the system, I think, is pretty clear. So you'll see neutrophils start to emerge, and you'll see a lot of them come through this megakaryocyte. And I'll call your attention to this field as they're passing through, because neutrophils are all sort of following each other, interestingly enough. So they're all going this way, and watch what happens right into space here. There, that. What was that? So what that was, was this. So here's a still of that image. So here's the megakaryocyte with its green membrane label. Here's a neutrophil, and it's trailing, it's like, looks like a comet. It's trailing this megakaryocyte membrane. And in fact, this happens pretty uniformly. So here's better images where we're better at it. Here's the neutrophil emerging, and it's taking green megakaryocyte membrane with it, and it's pulling this long strand of megakaryocyte membrane. So here comes others. Here's another one. Pulling a strand, this one's pulling a strand, this one's pulling a strand. So as they come and they leave, what they do is they go make this long, thin trail and then sort of pop off the end. So this stops the, there's no big continuity between the extracellular space and the megakaryocyte cytoplasm. But instead, at the end of the experiment, the megakaryocyte is now this big spider with all these, we call them megakaryocyte trails, that the neutrophils have pulled out of these cells. It's a really remarkable structure that emerges when these cells leave. And so you can see, again, here, the megakaryocyte, here, the neutrophil, and there's no neutrophil membrane on this. This is all a megakaryocyte membrane because there's no Lysix G, which is the neutrophil protein, but there's only CD41, which is the megakaryocyte protein. So and then these cells, this one looks to any self-respecting Star Wars fan like the Millennium Falcon. Even it said it's got this little thing here, just like this little thing here. So this is we call these Falcon neutrophils that have this part of the megakaryocyte that's trailing behind them. So the megakaryocyte is actually taking two things from the neutrophils, taking two things from the megakaryocyte, taking these intracellular dots, which we think are exosomes, and it's taking this membrane, which it's leaving behind in this trail. So what is this about? So we observe that the neutrophils elsewhere in the culture now start to attach to these trails. See how these neutrophils are all multiply attached here. And it turns out that this doesn't have chemoattractants that we can tell, but it has adhesion molecules of several different kinds that we can stain for, and see that the neutrophils seem to follow these, to track on them. And we wanted to see whether this is a way that neutrophils are marking their way along on endothelial cells. And so we generated a system in which we culture endothelial cells on a plate, add neutrophils that have become these, or that have gone through megakaryocytes and had the opportunity to become these falcon cells. And then we look by imaging. And so here is a neutrophil. Watch, she's got sort of this membrane stuck to it. This is a megakaryocyte membrane. Now it starts to deposit a trail. Didn't bring it from the megakaryocyte. The megakaryocyte is nowhere in the field, but it's bringing and leaving this long trail. Now it's coming back to interact with itself, its own trail, separates itself, interacts again, having difficulty leaving. Now it goes away, and here comes another neutrophil interacting with that trail. And here's another one interacting with the trail. Now this one starts to play with it. Where it's coming here? One of these starts to, this one here. Let me see if I got it right. This one starts to play with it, and actually makes it like kids playing with gum. It starts to drag more of this trail along. And so the neutrophils are making a kind of net out of megakaryocyte membrane that we think that they're using to label their own trail. And so, in fact, if you look at the other neutrophils and compare whether they're more likely to interact with random dots scattered around the image or with the dots that have CD41 on them, they're much more likely to interact with the ones that have CD41. So there's a kind of a documented interaction in this context. And so finally, we wondered whether this works in flow. So this is one thing to work in the culture dish, the other thing to work in circulation. So pending our 2-photon microscope experiments, which are quite challenging, we again took megakaryocytes with neutrophil cultures and add them now to a system in which you can introduce a flow on cultured endothelial cells. And you can watch what happens in this context. And so here is a video of one of these falcon cells migrating along. The endothelial cells are here, and here it sort of gets stuck, makes an attachment, starting to make one of these little trails. And then it's going to pop its way off and move on its way. Not so easily. It's sort of getting tethered by that. And then we can see that other cells are beginning to interact with these dots as well. So it turns out that the ones that are zipping along don't do it. It's the ones that are flowing that do in preliminary experiments. So the ones that are zipping through, they don't seem to attach. But the ones that are already in the rolling pathway, that is, they're going orthogonally or in some different direction to the flow, they do attach. So we don't think that the process is happening here in the capture stage. This is a Costley beautiful image of rolling, arrest, intravascular crawling, and transcytosol crawling. We think it happens here somewhere in the rolling phase. And these are all preliminary data. We need to quantitate them and confirm them by intrametal microscopy. Okay, so that's what I wanted to show you. And I'm eager for your thoughts and for discussion. So what we think is going on here is that the imperapolesis is going to be a way to modulate the pro-inflammatory capacity of neutrophils. And so the neutrophil enters, it absorbs exosomes from the megakaryocytes. It also exchanges membrane or takes membrane with it. It goes into the blood vessel, perhaps directly into the sinusoid. And then at some point when it starts to roll, it's willing to take this debris that's on the trailing edge of the megakaryocyte, of the neutrophil, sorry, and deposit it on endothelium, labeling where those cells, where it is left. And so we think that that's going to direct other neutrophils to the site. And this is a way for neutrophils to sort of lay tracer fire in terms of migration, although we have to still show that. Then when they get to the tissues, they have these exosomes in them. We know that they're better at migrating. We don't know for sure that they're better at other things. And that is something that we need to study if we can. So to conclude, I think there's a number of different concepts here that are new. So I think that we've defined through this the concept of an imperisome, which is this interesting vesicle that can go away, which is different therefore than a lysosome or a phagosome, at least as far as we can tell. We've defined this uptake of peptides and membrane, the Falcon neutrophils and MK trails, and the fact that this membrane transfer can transfer to platelets, probably also with proteins, although we don't know which ones, which enhances thrombopoiesis, but also has platelets that are different in the sense that they have other membrane on. And so since this interaction here is integrin modulated, my deep conviction that I hold without any supportive data is that it's going to be the activated neutrophils that do this, and therefore we might say angry neutrophils make angry platelets. And I think that's going to be true. I just don't have the data yet to show that it's true. So I think that adds imperapolesis to this axis of ways that megakaryocytes participate in immunity, but leaves lots of open questions. So what's the impact of imperapolesis on platelets? What is the impact on neutrophils in terms of their function? Is there, and how important is a trans-megakaryocyte route from or maybe back to the marrow? What are the mechanisms of imperapolesis? What are slow and fast imperapolesis and how do they differ? So what's that interaction with the nucleus all about? And are there other cells that do something similar? So is the presumption that cells don't go into the cytoplasm of other cells, is that true? Or is that some kind of a artifact of not looking hard enough? So with that, I'll thank, in particular, Pierre Cunin, who's been just an amazing person in the lab. Just got his K funded, will be promoted to assistant professor, and we'll have to figure out his brilliant future, and other members of the lab and some of our funding sources down here. So I will stop there, and maybe we can discuss.
That was awesome. It's kind of like science fiction, seeing how these cells interact. Does anyone have any questions? I've got a couple, but I'll just wait I don't know if you want to raise your hands or put something in the chat. I haven't actually looked if anyone's written anything in the chat. I mean, I'd never seen a picture of a megakaryocyte forming platelets. My sort of naive thought was that they just constantly produce platelets, not that the entire cell just disintegrates, basically, right, to make platelets. Once it's disintegrated, how long does it take does that individual cell recover? Or
Yeah, I don't actually know the answer to that. I believe I believe it does not. I believe, and if you look at lung, for example, you can see lots of sort of naked megakaryocyte nuclei. You don't see restored megakaryocytes. But I do not know that it could not happen.
And then just, any inflammatory process causes thrombocytosis. So you get more platelets. Do we know, is that because I'm sure somebody's looked at this. Are we making more megakaryocytes, or is it that we're more efficient at producing platelets on a per cell basis? In which case, is it because neutrophils are helping that process? Because it happens relatively quickly, right?
Right. So it happens quickly, but not overnight. And so the thrombocytosis, as we know, is a phenomenon of, 5, 6, 7 days into inflammation. And that is enough time to make more megakaryocytes from stem cells. And so it is thought that the pathway there is largely IL-6 stimulating TPO production that stimulates more megakaryocyte production. But I believe that megakaryocytes can also become more efficient in making platelets.
Yeah. One of the questions, have you ever seen another cell type besides neutrophils going through the megakaryocytes in that way?
Yeah, so
neutrophils.
Yeah, so we looked if you just incubate bone marrow with megakaryocytes in vitro, the first cell is neutrophils by overwhelming margins. The second cell is a B cell. And then way down the road are monocytes, T cells, NK cells. You can see them all, but they're really rare. What we haven't seen that in vivo much. So in vivo, if you look in the bone marrow, I don't know how often we've seen anything that wasn't a neutrophil. It's not common. So it's almost always neutrophils.
I was going to ask that mixing experiment that you did, the one that we were looking at, whether or not the megakaryocyte transfers to the platelet. I think you had a couple of mixing experiments. Did you ever? Yeah, I'm just trying to remember. It was the first half of the talk, because what I imagined you were going to do was do that same model and then either infect or stimulate that mouse. Meaning, if you do that, does the double labeled cell number increase? Right. So now you've got
production. Yeah.
Is there, because you showed that, I think earlier, that it's sort of, you drive that process by inflammation. So does the double?
I would be interested to see whether neutrophils that are activated do it better than not. The observation that we have that's somewhat relevant to that is if you look in if you look into a CD18 knockout mouse, so it's missing that pathway that at least partially mediates enteropoiesis. What you see is that enteropoiesis does no longer increases with inflammation. So it almost looks to us like there's a constitutive pathway and an inducible pathway. And the inducible pathway is integrin dependent and the constitutive pathway is not, or at least not so much. And that fits with this idea of angry neutrophils make angry platelets. So it's that you've upregulated a pathway that now allows the process to proceed. The challenge with this idea is that if you inject a CD18 knockout mouse with LPS or IL-1, which is I think the models we used, the mouse doesn't look that sick. So if you inject a wild type mouse with this, the mouse is hunched and cold and like it's miserable, right? So there's lots of other things that are not happening. And is it that the mouse just isn't sick enough? Or is it really that integrin is missing? And we don't have the answer to that. And so we didn't publish that data because we just don't know what the effect means. But I have my thoughts. So yeah, please, yeah.
So there's discussion. Sorry, yes. Neutrophils have a lifespan, and there's some argument that there are that once neutrophils get out into the tissue, they might last a little longer. Do you think these special class of neutrophils interacting with megakaryocytes lengthens their lifespan and allows them to basically be more effective effectors?
It's a really good question. And so it's one of the things we propose to look at. It has been harder than you might think to sort out the neutrophils that have done this versus not. And the reason for that is that megakaryocytes make microparticles. Microparticles are CD41 positive. They're really abundant and they stick to stuff. So if you look at a neutrophil and stain for CD41, you're going to see a large proportion of the neutrophils that you've cultured with megakaryocytes staining positive for CD41. And if you look by a microscope, that's probably, they don't all have this debris in them. It's probably just they have a lot of microparticles on the surface. So it's a technical problem we haven't figured out how to solve, to sort the ones that made it through and the ones that didn't. That's some technical issues that we have to get through.
Yeah, beautiful discussion. Very enjoyable.
Good, thanks.
Hans.
So I have never heard about megakaryocyte nets. That's a new concept to me. But neutrophil nets is a very well-known concept. And I wondered if you consider that there is any interaction between the nets from megakaryocytes and the nets from neutrophils. That trailing, and it all depends if your staining would show the nets of neutrophils.
So the nets are DNA and mixed proteins, and we actually don't see DNA in these megakaryocyte trails at all. So we often use DNA stain in these contexts, and we don't see any DNA in these trails. So we don't think that they're nets. We think that they are a membrane tubular structure. And so we haven't seen that the neutrophils that pass through evidently net more than others, but that's a hard question and we haven't examined it carefully enough to say yes or no. We haven't tried to see what happens if a megakaryocyte is exposed to neutrophil nets. But I would guess that that's not a common situation in vivo. Although I don't know what happens when they hit the lung, and maybe they might hit. We think in sepsis that there's some degree of netosis that happens in tissues, including in the lung, and that's one of the bases for ARDS. I'm talking beyond my knowledge here, but I think that's possible. And what happens when the megakaryocyte hits a net? I don't know. It's a good question.
Bruce.
Sorry for asking all these questions. You indicated
It's awesome. That's a great discussion.
So you argued that agri-neutrophils probably seek out megakaryocytes. That indicates that they had to be activated somewhere by something and specifically seek out. Do you think that they come out? Are the megakaryocytes in the same tissue? Do they come out of the tissue? How would you envision this happening? I think it's an intriguing, statement that you made.
I have two ideas. The first is that I would guess if you looked at bone marrow neutrophils in a mouse that did or didn't have an LPS injection into its abdomen, you would see that the neutrophils are more activated in the bone marrow just because there's LPS around. So I think a septic mouse probably has neutrophils that are partially activated in the bone marrow. I have never done the experiment, but I wager that it's true.
How about a more localized infection? Something that's not a systemic infection.
Yeah, it could. We have seen imperapolesis in the spleen, just as an extramedullary hematopoietic organ, so we've seen it there. We have not ever visualized in the lung, and visualizing lung megakaryocytes is really hard. We did now. Pierre did go to the Looney group and we're just waiting for the apparatus to visualize the lung, and we're hoping to see with a little more detail what goes on in that context. But we've never seen imperapolesis happen in the lung so far, but we've had very little opportunity to see it.
Thank you.
So there's one possibility is that they're assuming that most of this happens in the bone marrow. The idea would be that neutrophils in the bone marrow get activated by circulating mediators. That's one. The second is we know that neutrophils return to the bone marrow many of them return once they're done. Now, a lot of them die in the tissues and get phagocytosed, but others return either to the lung or to the bone marrow. And this is mediated through a CXCR4. what neutrophils express as they get older. So when they return, neutrophils bring information from the periphery back to the bone marrow. And it is possible that that information is just wasted and they're killed and eaten. But since nothing is wasted, it seems like that information must in some way be processed. And I'm going to go here a little further, since we're just speculating wildly. When megakaryocytes, if you watch these beautiful Uli van Andrian videos of megakaryocytes making platelets from the calvarium, what you see is that they reach these proplatelet fingers into the circulation, into the sinusoids, and those get gradually sheared off, right? But for a while, you have a cell that has feelers into the blood. And so again, that cell is sensing what's going on in the blood. Megakaryocytes know, the whole body knows what's happening in the blood. And I think that information is doing something. And so it may be that those feelers are grabbing cells in the circulation, right? That's one possibility that one could test using in vitro microscopy. Or it could be they're sensing soluble mediators and sending that signal back to the mothership. Or it could be that nothing like that is happening at all, and it's just really just simple shearing off fingers.
Interesting. So a sensor cell.
Yes. Yeah. It would make sense to me that this could happen. I think mast cells have some evidence that they do this, that they reach, the mast cells have fingers into the vasculature. And so maybe megakaryocytes are just even better at that than mast cells.
Interesting. Thank you.
Adrian can tell us about that
So I have a related question. So because there are lots of megakaryocytes in the lung, so that was interesting that we don't really see this happening in the lung. So does that mean that you need certain stage of the megakaryocyte has to be at certain stage that this would happen?
So we know in vitro that if you differentiate, as you're making megakaryocytes, day 1, day 2, day 3, day 4 of culture, the day 4 culture ones do it a lot better than the day 3 or day 2 or day 1 culture cells. So there is a maturational benefit in terms of empiripolesis. So lung megakaryocytes remain somewhat controversial, I think. There's a paper from Craig Morell's group in Rochester. There's a paper from Mark Looney's group that part of this Nature paper was sorting megakaryocytes from lung and identifying 2 different phenotypes. But in neither of those, in the Nature paper, there was no purity data at all. No flow cytometer gating, no imaging, nothing. And in the Morell paper, they use mostly PF4. But PF4 is not specific. There's subsets of macrophages that express PF4 as well. And so, are those megakaryocytes, are they not? I don't know. But if you look at mouse sections for megakaryocytes, you don't see very many. And when you do, if you look at that video, you see them come in, they pop, and they go away. So we will hope to see better when we start to do some intravital imaging using PF4 CRE. So they have cells lighting up in the lung and see which cells light up as PF4, and then also a PF4-GFP. And we'll hopefully image the megakaryocytes directly. And we'll be able to say, do we believe that there are interstitial megakaryocytes in the lung, or do we think that there aren't?
Thanks.
So why do megakaryocytes have this unique ability to have cells transit through them when the receptors you're talking about are on lots of cell types? What allows that to happen? I guess the initial entry, and then it was intriguing that the membrane, the extra membrane in the megakaryocyte might have some role in the sort of exit from the EP, I guess. And so it does seem like science fiction, right? Like it's against all our rules. Like we don't have cells passing through cells. That's not what we think.
Well, we we didn't expect to see this when we did the EM. That was not what we thought we would see. So I think that we don't often do EM of this, and so this kind of thing. So I think that it could be that it happens and we just haven't noticed it. It is also possible that the main, you might say, raison d'etre for this process, if it involves transfer to the DMS, that's a megakaryocyte specific phenomenon. Other cells don't really have that structure. So maybe other cells don't need it. It's not the fact that the surface receptors are different, it's that somehow otherwise there's a machinery that allows this to happen. And CD18 is not sufficient to have, it's neither necessary nor sufficient to have in peripoiesis.
But the way
but
I
I
understand
also
it,
say-
peripoiesis is the actual in the vacuole, right? And then, I mean, I guess it's the whole process that you described, right? Because it's free moving cell. Like when you think of
Right.
EP, I had to look up EP yesterday to be able to.
Yeah,
Like,
yeah.
EP's easier, so I'll say that. But
It's easier, yeah.
what's like when you think of that, you're thinking about the whole process, the entry and the free movement in the cytoplasm. When that's what the initial observations were, is there like free moving cells within cytoplasm?
Yes, we think it's the whole process, and we think that the whole process of entering, moving through in a way that doesn't kill the cell. So it's different from phagocytosis, where the cell dies, from entosis, which is a similar process in which sort of ends up in cell death. And we think it's a distinct kind of cell-in-cell interaction.
But wouldn't a neutrophil like be able to punch its way into lots of cells, but then maybe they wouldn't get out of the initial
Right.
entry, right?
So I think, so it's a really good question. So we have never actually tried, say, to differentiate megakaryocyte macrophages to be really big macrophages and have them feed
They're
the
like
neutrophils.
they're equally big,
what
right?
happens.
I mean, there's lots of room for them to.
Or, I don't know if they're equally big, but like a giant cell is certainly as big. So could a giant cell do this? But we don't see giant cells with cells in them. This phenomenon of peripoiesis is well established in histopathology circles, but other cells don't seem to do it. That said, though, if you remember that first video, the first still of a bone marrow of a mouse with lots of neutrophils, here's some megakaryocytes. I mean, if you were just looking at that, you'd say those cells are on top, right? Those cells aren't in. You would just assume that. So, one example of this it's sort of related phenomenon has to do with the role of microparticles in synovial fluid. So, generally, if you flow cytometry synovial fluid, so from inside a joint, you then gate on the cells, right? And you throw everything else away because it's all debris, right? But it turns out that most of that debris is incredibly abundant microparticles. Similarly, in blood. You throw the stuff out on the flow cytometry gating that's not a certain size because you think it's irrelevant. But it's not irrelevant. It's exosomes and microparticles in blood that are incredibly abundant. And in fact, the most abundant source of microparticles in blood, they're CD41 positive. They're megakaryocyte microparticles. They're not even thought to be platelet microparticles. They're megakaryocyte microparticles. So
that's kind of brings me to my second question, which is,
Yeah.
because we work on Wiskott-Aldrich syndrome patients, and so they have microplatelets, and I would assume they have a lot, like proportionally a lot more microparticles. And do you think that has any role in the inflammatory status of Wiskott subjects? Like just the fact that you're making more microparticles?
So I
hadn't
have
really
some-
thought about that, but it seems like there's a
Yeah.
lot, right?
I think it's a reasonable thought that do they have more circulating microparticles? It's not that hard a question to ask. You have to make sure the platelets don't clot in your preparation, in which case they will release a lot of CD41 microparticles and make it all a mess. But if you make a good plasma, it should be easy to do.
Yes, we don't know, but the presumption is they probably would, because they have these because the process of platelet generation is so disrupted.
Mhm. I know zero about Wiskott-Aldrich. And so I don't have any opinion on what would happen in the platelet compartment or megakaryocyte compartment in that condition.
I was waiting for Hans to ask that question. I was assuming it was
So
going to
I was thinking about it, but the Visigoths have a problem with actin generation, with actin polymerization. And I saw in some of your slides that actin is considered as part of this interaction. Is that true? And could we use these models like Viscot that have this problem with microplatelets and actin polymerization? And otherwise, we have patients, like Grimm syndrome, where this migration of cells, which you mentioned, from out of the bone marrow into the periphery and back in, is not possible because of that defect of the neutrophils. And third, if you have neutropenia, would that not then interfere with platelet production or with the number of platelets? And I have never seen that in completely neutropenic patients, that they also have thrombocytopenia.
So that would have been nice. We tried that experiment to deplete neutrophils and to see whether the platelet numbers go down, and they don't. Similarly, we've depleted megakaryocytes using, I think I showed some of this data earlier to Eric, but we depleted megakaryocytes using a TF4 DTR system with recurrent injections of diphtheria toxin, and we don't see any problem necessarily with release of neutrophils from the marrow. So, I think that I think if the answer were really simple, we probably would have hit on it already. I don't think it's going to be that simple. We don't have a blocker of imperapolesis. The only blocker that really works is if you block the actin cytoskeleton, which is not a viable strategy in a living mouse. Nor specific. So if there is do you think that we could use the Wiscott's? There are Wiscott mice, right?
Yeah.
Model mice.
Yep.
What's that? So
yeah, they're a little different. Their thrombocytopenia is not as pronounced as in humans.
And do they have imperapolesis? I don't know. How bad is their actin defect? I mean, if they had bad actin, they would die, right? I mean, there would be.
It's a partial defect. It's a partial polymerization defect.
It would be very interesting to see whether they have less imperapolesis and fewer hybrid platelets, although that's a really hard thing to test.
Well, we have the mice, if you want them. Yeah.
Well, actually, just bone marrow sections would be fine. We could probably just look at some bone marrow sections and see if we have imperapolesis or if it's totally absent. We need enough to the prevalence on 5 micron sections of 2 to 3%. We would need to see a good number of mice to see less. But if it were 0, that would be the first system I know that's described that has no imperapolesis.
Mm-hmm. It's probably not 0 because it's not a complete defect, but certainly we could check. I don't know how many mice we have. We only studied the B cell side, so we use them as donors for chimeras, and then you get lots of recipients with one donor. But we definitely have animals.
Maybe save some bones for us.
Another interesting model would be leukocyte adhesion defects. And there are animal models. I know there are dogs. I guess there are mice. And these are patients who have no neutrophils in the periphery, but they have plenty of neutrophils in the bone marrow. So they would not be able to interact with megakaryocytes in the periphery, like in the lung, but they should be able to interact in the bone marrow. Actually, they have neutrophils in the blood, but not in the periphery, not outside of the blood.
Yeah, they can't exit.
Yeah, they can't exit the blood.
Yeah.
So they have this neutrophilia. Yeah, but CD18 knockout mice, are an LAD version, right?
Yeah. But there are 3 LADs, and one has thrombocytopenia associated with it. These are patients, Amos Etzioni would have them. They are mainly in the Middle East and no consanguinity. So they are very interesting in point of view. They have no neutrophils in the tissue, and they have thrombocytopenia and bleeding.
Hans can find you a patient. He knows the name, I'm sure. All right. Well, I think we've gone well beyond your time, and I think you're about to. We should probably give you 5 minutes break before you meet David Rawlings. Thanks a lot. That was really cool.
Thank you.
Thanks all. Thanks for the invitation.
Sure.
Bye.